Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. We are on session 4 where we are going to discuss the questions on electron transport system and C3 cycle today. Okay. So, yes, already we have solved half of the questions from electron transport system, that is ETS of non-cyclic photophosphorylation, quite a few questions from cyclic photophosphorylation as well. Now, we are going to exercise the C3 cycle as well along with the electron transport, that is non-cyclic photophosphorylation. As we have discussed the concept in detail yesterday, we are directly going to solve the questions today. Okay, Vita? So, you look into this question, study the given flowchart of cyclic photophosphorylation. He is saying cyclic photophosphorylation. We have discussed yesterday, right? Cyclic photophosphorylation will be operated in photosystem 1, which is in stroma lamellae. Okay, this is present in stroma lamellae, which is in PS1. Okay, only photosystem 1 is involved, which is in stroma lamellae, where it will conduct an oxygenic photosynthesis. You can also call it as bacterial photosynthesis. So, here you see the light energy which is being fought on the chlorophyll. So, here the cyclic photophosphorylation from this image, what is the question that is asked to us? To label A, B and C. Okay. First you observe carefully that A will be obviously photosystem 1, right, because only PS1 will be involved, and B is what? Electron acceptor. Okay, B will be electron acceptor, it is not proton acceptor, it is electron acceptor, and C will be what? Reaction center, reaction center. What is the maximum absorption capacity of photosystem 1? P700 nanometers, right. So, it will be, this reaction center is at P700. So, now you look into it carefully, Vita. So, A is what? PS1 and B will be electron acceptor, C will be P700. So, here the option is B. Clear? Next question you see. Yeah. Identify A, B, C, D. So, here the question is from non-cyclic photophosphorylation. Of course, here the light harvesting complex which is associated with photolysis of water in the inner thylakoid membrane. Okay, in the inner thylakoid membrane, if it is associated with photolysis of water, then this light harvesting complex belongs to what photosystem? PS1, sorry, PS2. Okay, this light harvesting complex belongs to photosystem 2. So, here you mark A, where A is photosystem 2, C and D, just encircle that and keep. Now, you observe this electron acceptor at B. B is what? Electron acceptor, this is not a proton acceptor. Fine, the continuous supply of electrons from light harvesting complex from the core, from the antenna pigments to the reaction center, the continuous supply of electrons is being passed out from what? Photolysis of water, where electrons are being submitted to what? This reaction center, that is B. This is an electron acceptor. Now, you look into C. C will be what? Electron transport system. This is electron transport system. And then D, this is what? PS1, photosystem 1, this light harvesting complex which is not associated with photolysis of water is what? Photosystem 1. And here E, the ultimate electron acceptor will be NADP plus. Accepting the electrons, NADP plus will be reduced to NADPH plus H plus. Clear, right? So, here what answer we have to mark? Option D. Clear, ma? So, you look into it carefully once again. First, A will be the light harvesting complex which is associated with photolysis of water, then A will be PS2. This B is what? Electron acceptor, that is not a proton acceptor because electrons are being transported. And C is what? Electron transport system. This light harvesting complex which is not associated with photolysis of water is what? Photosystem 1, and the ultimate electron acceptor is NADP plus which is reduced to NADPH plus H plus. Clear, right? So, here the option will be D, beta. Next, the Z scheme of photophosphorylation follows what scheme? Okay, Z scheme is following what sequence he is asking. Definitely it will be from PS2 to electron acceptor. Okay, first from photosystem 2, if this is the thylakoid membrane, from photosystem 2, the electrons get excited and they are submitted to what? Reaction center. This reaction center is what? Reaction center is electron acceptor. This is what movement of electrons, ma? Uphill movement of electrons. So this movement is uphill movement of electrons, and this continuous chain from the reaction center to primary electron acceptor, that is pheophytin, to last electron acceptor of PS2, that is plastocyanin. The electron transport is what, Nana? Downhill. This is downhill. And from here again electrons are excited and given to the reaction center of PS1, and this reaction center is at P700, and here the electron movement will be again uphill. Fine. Again lastly downhill movement. Okay, again a downhill movement of electron transport system. Have you understand this? Uphill, downhill, uphill, downhill. Uphill, you just simply remember beta, from photosystems to the reaction center, the electron transport will be uphill. To the protein molecules, the electron transport system will be downhill. That's it. Now you solve this question, it will be easy for you. So from PS2 to the electron acceptor or reaction center, it is uphill. So you encounter or mark these two options, A and D. So first it is uphill. From that reaction center or electron acceptor to photosystem 1, it is downhill transport. Okay. Again from PS1 to the reaction center or electron transport, that is uphill movement. Again to the ultimate electron acceptor or the final electron acceptor of the non-cyclic photophosphorylation, it will be what, Nana? Downhill movement. So here what will be the right answer? Option A will be the right answer. Clear? Next question you look into, ma. Yeah, this question is from Kelvin cycle. Find the number of ATP required for reduction step to produce one glucose molecule. So before solving this question, and now we are going to solve few questions on the C3 cycle or dark reactions as well. So let's exercise the concept once in brief. So light independent reactions. So what are the light independent reactions? Light independent reactions are also known as dark reactions because it is not taking place in dark, but those are purely dependent on the products of the light reaction, not on the light. So light independent reactions are dependent on what? Products of, they are purely dependent on the products of light reaction. What are those products, Nana? ATP and NADPH. So utilizing this ATP and NADPH, carbon dioxide which is accepted to the leaf surface. Okay, carbon dioxide which is accepted through the stomata into the mesophyll cell, which enters the stroma of the chloroplast. This enters what? Stroma of chloroplast. Fine. So this carbon dioxide is being fixed or reduced to glucose, C6H12O6, by utilizing what? ATP and NADPH. This reduction process is different in different plants. So based on the plants, type of plants where the reduction is happening, if it is tropical plants, C3 cycle is activated. Okay, C3 cycle is activated in all the plants, whether it is tropical, temperate or desert plants. In all the plants, C3 cycle will be present. Okay, C3 cycle is activated in all the plants. And one more cycle, that is C4 cycle, is activated only in the temperate plants. Okay, they are activated only in the temperate plants. CAM cycle is the third cycle. This cycle is observed, CAM cycle is observed in the desert plants. So remember this, ma. This is very simple, right? From basic level I am saying you. See, So, light independent reactions, which are also called as dark reactions. These dark reactions are dependent on what? Products of the light reaction. What are the products of light reaction beta? ATP and NADPH. Utilizing this ATP and NADPH, carbon dioxide which is present in the stroma of the chloroplast is fixed to glucose. Okay, carbon dioxide is fixed to what? Glucose. This fixation is different in different plants. C3 cycle is activated through C3 cycle. If glucose formation through C3 cycle happens in all plants, but there is C4 cycle, this is activated only in the temperate plants. In only temperate plants C4 cycle is observed, and CAM cycle is seen in the desert plants. Clear, right? You have understood this. So now let us look into C3 cycle only today. I will tell you the gist of C3 cycle. You observe, let's suppose this as a leaf. Okay, this as a leaf, and here carbon dioxide has entered into the mesophyll cells. Inside mesophyll cells we know chloroplast is present, and in the thylakoid region it will not enter. So the carbon dioxide which is present in the stroma, one carbon dioxide is accepted by RUBP. Okay, this is ribulose bisphosphate, which is 5 carbon compound. One carbon dioxide, what is the primary carbon dioxide acceptor? RUBP is the primary carbon dioxide acceptor. And in the presence of the world's abundant enzyme, which enzyme is helping out in this reaction? World's abundant enzyme, that is Rubisco. Fine. This Rubisco, after the reaction, it forms 2 molecules of phosphoglyceric acid. 2 molecules of phosphoglyceric acid. You can count it beta. This is 5 carbon and this is 1 carbon. Total 6 carbon. So 2 molecules of 3 carbon compound is formed. This first step is called as carboxylation. This first step is what, Nana? Carboxylation. Second step, which is reduction step. Okay. Second step is reduction step, where 2 molecules of phosphoglyceraldehyde is converted to 2 molecules of glyceraldehyde 3-phosphate. Okay. 2 molecules of phosphoglyceric acid is converted to 2 molecules of glyceraldehyde 3-phosphate. This is the reduction step, wherein 2 molecules of NADP plus NADPH and 2 ATP are utilized per 1 carbon dioxide. Do remember, per 1 carbon dioxide how much NADPH and ATP are utilized? 2 NADPH and 2 ATP. Fine. So if this is being given, it means it is utilized. If this energy assimilatory power is over here, that means it is released. Remember, most of the students will get confused whether the energy is over here or here. If it is over here, it means it is utilized. If it is over here at the end of the arrow mark, it means in this reaction process that the energy is being released. The energy is released during aerobic respiration, that we will discuss in detail in the next chapter. Now you see this 2 molecules of glyceraldehyde, which is on total it is having 6 carbons, right? Of this 6 carbons, 1 carbon will be escaping from each Kelvin cycle to form 6 carbon glucose. 1 carbon will be escaping all the time to form what? 6 carbon glucose. 1, 2, 3. Okay. So as such, the rest 5 carbons, with the remaining 5 carbons will be regenerated back to RUBP. For this regeneration step, so the third step is what? Regeneration step. For this regeneration step, 1 more ATP is being utilized. Till here it is clear, na beta? There are further 7 more steps in reduction that are not required for NEET. Okay? Till here it is enough. So you look into it. 1 carbon dioxide by taking RUBP, which by accepted by the primary acceptor RUBP, in the presence of world's abundant enzyme Rubisco, it is getting fixed as 2 molecules of PGA, phosphoglyceric acid. This is being reduced to 2 molecules of glyceraldehyde 3-phosphate by the energy 2 NADPH and 2 ATP. This is getting reduced to 2 glyceraldehyde 3-phosphate. Okay. this glyceraldehyde 3-phosphate, of that 1 carbon will be escaping Kelvin cycle to form the glucose from each Kelvin cycle. The rest 5 carbons will be regenerated. Here how much energy is being utilized for 1 carbon dioxide? 1 carbon dioxide on total 2 plus 1, 3 ATP and 2 NADPH are utilized. But when only 1 carbon is being escaped from each cycle for formation of glucose, as glucose is 6 carbon, 6 times the Kelvin cycle have to run. When 6 times the Kelvin cycle have to run, how many carbon dioxides are required? 6 carbon dioxides are required. How much ATP will be required? 6 into 3. Okay, 6 into 3. How much ma? 18. 18 ATP will be required. And how much NADPH? 6 into 2, that is 12 NADPH. NADPH will be required. If the question is for the formation of 1 glucose, how much energy is being utilized only in the reduction step? Only in the reduction step. Only in the reduction step for 1 carbon dioxide it will be 2 ATP. For 1 glucose or hexose molecule, as 6 carbon dioxides are required, that will be 12, 6 into 2, right beta? 6 into 2, it will be 12 ATP. Clear, right Nana? Easy, right? So this is all about the Kelvin cycle. Now here you see, let us exercise the questions so that it will improve your application. Okay, look into it here. Find the number of ATP required for the reduction step to produce 1 glucose molecule. Just now we have discussed, right, that is 12 ATP. Okay, now you see this question, an application question. You see, a photosynthesizing plant is releasing 180 more than the normal. Okay, 18 oxygen. This is an isotope, sorry. Okay, this is the figure. So this is an isotope, oxygen 18. Okay, so this isotope is released. Okay, in a photosynthesizing plant is releasing this oxygen isotope, oxygen 18 isotope more than the normal. The plant must have been supplied with. Firstly, this is very simple. You have to know what are the raw materials for photosynthesis. That is from the reaction of photosynthesis. Okay, most accepted equation of the photosynthesis, that is 6CO2 plus 12 H2O in the presence of light C6 H12O6 plus 6 H2O and 6 O2, right? This is being evolved as byproduct. So what are the raw materials here? Carbon dioxide, water and sunlight in the presence of chlorophyll, right? So these are the raw materials. Along with this, if these are the raw materials, then you cannot see a O18 isotope. Along with this raw materials, if there is O18, then only you can see O18 byproduct in the products of photosynthesis. Understood beta? It is very simple. First you have to know what are the raw materials. Along with this raw materials, O18 have to be there. Then only at the product side you can get oxygen 18 type of isotope from the photosynthesizing plant. So here what is it supplied with is the question. Definitely it is not with O3. Water and O18, there is a possibility. Definitely it is not with glucose and oxygen and carbon dioxide and O18. Without water, if just CO2 and O18 is supplied, how is that producing O18 at the end product? That is wrong. So it is not compensated just with carbon dioxide and O18. So what option we have to choose beta? Water and O18, then only you can see this type of isotope at the end products, as normal oxygen is being released from what? Water. Normal oxygen is being evolved as a byproduct from photosynthesis is from water. Clear? So this is about this question. Yeah. This question you look into ma. Photolysis of water requires what? Photolysis of water requires light, that's it, sunlight, because utilizing sunlight only water molecule is being split into 2H plus 2 electrons plus oxygen atom. Okay. So, photolysis of water utilizing the light as the photons, utilizing light energy as photons, water molecule is split into 2H plus 2 electrons plus oxygen atom. Clear, right, Nana? So, this is light. Fine. Next, ETC, electron transport chain of photosynthesis process is, okay, ETC of photosynthesis is what? This is bound to the thylakoid membrane, that is correct only. It is present in stroma, wrong, because electron transport chain is operated inside the thylakoid membrane or thylakoid lamellae. This is present in stroma, no, it is not present in stroma. This is bound to the outer chloroplast membrane, wrong, this is not bound to the outer chloroplast membrane. See, if this is the chloroplast I consider, we all know chloroplast is a double membrane bound organelle, right, ma? Chloroplast is a double membrane bound organelle. Inside a chloroplast, membranous sac-like structures are present. Okay, inside chloroplast what is present? Membranous sac-like structures are present. This membranous sac-like structures are connected through stroma lamellae. Fine. So, if I take this one thylakoid membrane, over this thylakoid membrane, the Z scheme will be operated. Inside the inner mitochondrial membrane, photolysis of water takes place. Now, you see ETS is operated where? Inside the thylakoid membrane, not in the chloroplast membrane. Dispersal in cytoplasm, no. So, ETC of photosynthesis is bound to thylakoid membrane only. Okay. Whenever you are not getting any question, convert that question into a diagram point of view, you can easily answer. Okay, na beta? Next question. This you look into, recognize the figure and find out the correct matching. Okay. So, this question is from what? Chemiosmotic hypothesis. Chemiosmotic hypothesis. So, here you see ma A, okay. First, this will be what? ATP synthase, that we all know, right. So, this is what? ATP synthase, where this is F0 and this is F1 complex. So, this is labeled as what? F. So, F is what here? ATP synthase. Just look into the options. F is not the lumen, so you can cancel it here. F is not the stroma. Yes, yes. F is stroma. F is stroma. And here D you can see, right? D is what? ATP synthase. So, D is ATP synthase and F is stroma. This you can opt. Next, A is photosystem 1. Wrong. A will be what? PS2. PS2. Okay, not photosystem 1. So, this also you can eliminate. Here you see ma F is stroma, D is ATP synthase and A will be photosystem 2. True. And B is photosystem 1. B is photosystem 1. And C, if you have to see, this is after photosystem 2, inside photosystem 2, after excitement of electron in ETS, just recall what are the electrons that are present from reaction center of P6E80. The primary electron acceptor is pheophytin. After that it is plastoquinone. Then PQH2. H2 will be directly given to what? Thylakoid lumen. From here to cytochrome B6 F complex. Next to what? Plastocyanin. So, this will be the electron flow. So, in midst that what is CMA? Cytochrome B6 F complex. So, here we can go with option C, okay, wherein this is what? ATP synthase and F will be what? This complete fluid filled space is stroma. This is PS2, PS1, cytochrome B6 F complex. Clear beta? So, this is from the chemiosmotic hypothesis. Next question. Yeah. So, before going to next question, I would like to end up this session by giving you a gist or clear explanation on chemiosmotic hypothesis, so that you can solve any question that is coming to you from chemiosmotic hypothesis. Look into this beta, chemiosmotic hypothesis. There is one paragraph in your NCERT. Many of you will get confused that chemiosmotic hypothesis is explained in 3 paragraphs, right? So, firstly, how the chemiosmotic hypothesis is operated when in thylakoid lumen, because of photolysis of water, more protons are present in the thylakoid lumen, where less protons will be present in the stroma. Okay, in stroma H plus are less, whereas in thylakoid lumen H plus are more. As such, the proton gradient is operated in between thylakoid lumen and stroma. What are the main reasons for this? The very first reason, in PS2, in photosystem 2, when the electrons are being accepted by the pheophytin, after that plastoquinone, right? So, this plastoquinone, rather than it is called as an electron acceptor, this is a proton acceptor, where the protons are being accepted from the stroma are being taken by plastoquinone, and this will be converted to PQH2. This H2 will be sent back to thylakoid lumen. As such, protons are being taken from the stroma and given to what? Thylakoid lumen. The very first thing is, due to photolysis of water, more protons are present inside the thylakoid lumen, and the second reason is, during electron transport chain from plastoquinone, again protons are being grabbed from the stroma to thylakoid lumen. Third reason is, the ultimate or final electron acceptor is what, ma? NADPH, NADP plus. This is the ultimate electron accepting 2 electrons in the presence of the enzyme NADP reductase. In the presence of the enzyme NADP reductase, it is converted to NADPH plus H plus. Clear? It is converted to NADPH plus H plus. So, this is being reduced to NADPH plus H plus. So, because of these 3 reasons, the proton gradient will get completely lowered in the stroma, and you can see high proton concentration inside the thylakoid lumen. Clear, right beta? These are the 3 reasons.